The next speaker, Dr. Haung from Columbia University from New York City, is talking about how you can actually remove tau from brain cells. I hope that is correct in a way.
Yeah.
Okay, thank you.
Thank you very much, Alex. I wanted to thank Robin and the Brain Support Network. I think it's a great opportunity for scientists and research and clinicians to kind of speak with individuals who are affected by diseases like frontotemporal PSP and CBD, and to kind of give an idea of what our research entails and what it means in terms of the disease. And my apologies, my voice is actually also going down, so if I do start to slip, just kind of scream out again and I'll try to raise my voice again. So the title of my talk is kind of removing protein buildup in your brain cells. And while we kind of talk about tau as being this pathological cell constituent, it actually also has a specific purpose. And so we want to, so in my talk, what I'll do is discuss how we can remove it, but I also want to kind of give a little inference as to that it does have a function in cells normally, but it's when it becomes disruptive, that's when you want to remove it. So we'll talk a bit about how one could go about this process. And what I'll discuss is something that I work on in my lab called autophagy. It's Greek, two words from Greek, auto and phagos, which is self-eating or digestion, self-digestion, and why it's important in brain cells to activate this, and how we might be able to regulate this using drug development to improve the whole process, and hopefully what that means to the disease treatments in the future. So before I start with my talk, I wanted to kind of identify my funding sources, as well as a biotech company that's formed around some of the technologies and drug development that we're pursuing in this avenue. So tauopathies actually cover a lot of diseases, and in fact actually there's over 20 different neurodegenerative disorders, and of course we're here to discuss progressive supranuclear palsy, cortical basal degeneration, but there's others like frontotemporal dementia, PICS, as well as Alzheimer's disease, and something that's becoming quite important in the news is traumatic brain injury and chronic traumatic encephalopathy. And so what we can do is actually study all these different diseases using the context of tau as the central point, where we can start to kind of go, well, if we can remove tau, does this not only help the disease process, disease reversal in PSP or CBD, but all these other disorders? And so we kind of use this as a focal point for discovery and as well as translation into medical treatments. Tau was originally described, and it wasn't really just known as tau back then, but in these upper panels, these are images, and magnifying glass. These are images, actually drawings from Alzheimer. And Alzheimer, of course, name sounds familiar. He is the one whose name now is part of Alzheimer's disease. So he originally described the disease process. And one of the first things he noted was these cells, these neurons, had these very kind of like filamentous string-like structures in the brain cells. And they call them neurofilament tangles. And you can actually see an image of how we look at it now using antibodies that stain for tau. Now, tau normally is a protein that helps the cytoskeletal. So that cytoskeleton forms that structure and also allows for transport within the cell. And that's our best guess is what tau does. It helps facilitate some of that function. But as I said, sometimes it gets disrupted in brain cells. And here we've got images from a paper from Dennis Dixon, who's one of the foremost neuropathologists in the field of cortical basal degeneration, as well as progressive supranuclear palsy. And you can actually see brain cells called neurons staining here, as well as some of the what we call the astrocytes or glia, which are important for kind of the immune function of the brain cells. So it disrupts multiple different brain cell types. And what we want to try to do is see if we can reverse that process, take away that buildup of these types of kind of aggregated forms that start to appear in the disease process. And my lab is kind of focused on getting rid of the garbage in brain cells, and that's really the best guess. And when I tell that to my family, they're kind of like, so you're a garbage collector? And I said, if it helps, then that's fine. We have a couple of ways in all cells to kind of get rid of garbage, and the first one is kind of what we call the proteasome system. And the proteasome system kind of gets rid of all the little bits. It's sort of like a Pac-Man that gets rid of some of the little specks that are in the road, but it doesn't get rid of the big clumps. And that's what we use autophagy for. And so in this cartoon, we've got tau, and basically tau can kind of be removed by several processes, including the proteasome system and the autophagic pathway. And they kind of work in concert together. So normally a brain cell is really efficient. It shouldn't kind of build up a lot of garbage. But with aging and with disease, it does happen. And so what we want to try to do is figure out, well, what happens with these systems that they start to decline in age and disease? And is there some way that we can kind of boost them to really kind of ignite the whole process of what we call basically clearing out the garbage? And so I kind of break it down into a fairly simple term. Brain cells do build up garbage. It happens. And what we want to do is probably in this autophagic pathway is kind of think about these autophagic vacuoles, which basically collect the garbage. They have to travel down these roads and bring it to a recycling center. And it's not a garbage dump, it's a recycling center, because what you do is you take those tau proteins or other proteins that start to build up, or lipids or sugars, and you break it down so that the cells can rebuild. And so this is why we call it a recycling center in terms of some of the utility. Now, part of the process of identifying where disease takes place and what part of this pathway seems to be disrupted is really an important avenue of research in neurodegenerative diseases, including PSP, and something that I've been kind of focused on for the last decade of research. And we know, going back in some of the historical stuff, is that we have what we call these autophagic vacuoles, and these are these autophagic vacuoles. When I first started out, they didn't think they actually occurred in brain cells, in neurons, and it was because they didn't see them. And the only way we could see them is actually when we start to disrupt the brain process. We disrupt something we call the lysosome, which is kind of that recycling center. It breaks down the garbage that is delivered by these autophagic vacuoles, which basically envelop the garbage and they transport down and deliver it to the lysosome where they can get rid of them. So somewhere in this pathway, we thought there's some disruption. We know in early stage diseases, the garbage collection is not happening fast enough. And so some of the models have suggested that, well, if we kind of boost this kind of garbage truck collection, maybe that'll help the whole process. And that does to a certain point. But we know later on disease that some of these processes become a little bit more disrupted. And so we want to try to improve the transport, these roads that kind of are built in the cells. Or maybe we want to improve the garbage truck entry into the lysosome or the recycling center. And finally, the recycling center itself is known to be disrupted or dysfunctional in various diseases, including Alzheimer's disease. And this is a brain cell from a patient who had Alzheimer's disease. And what we were able to see is there's something called dystrophic neurites. And so if you kind of see these processes, these are brain cells, and these processes in Alzheimer's disease will start to build up these kind of lumps, like this one here. And what those are turned out to be actually these dystrophic neurites, and they're filled with these autophagic vacuoles. So we know that it's not getting to where it needs to get to break down the garbage. And that itself becomes damaging because basically it creates a physical barrier for anything else to be transported on. And one of the research avenues that we looked at is sort of like, can you see basically clearance of garbage and where garbage builds up, and is there a coincidence on that? In some of the early work that we've seen, we see that in cells that have the garbage buildup, it may not be so efficient. So this green kind of marks where these autophagic vacuoles would be. And you can see the red is right at the top of where the brain cell is, almost like what we call the cell body of the cell, or what probably is more like amorphous to the head of the cell. And we can kind of see that some transport's taking place, but it probably isn't being efficient enough. And this is in a mouse model of tauopathy. And what we want to try to do is improve this whole process. And some of my research has been kind of focused on how do we improve that latter process, because we know that a lot of the treatments probably work a little bit earlier on disease, but with devastating disease, it can happen very quickly. And we want to make sure that we can kind of improve that process faster. So I've kind of focused on a process where we basically want to start to improve this clearance mechanism through a pathway where we try to activate an enzyme that increases the fusion or increases these autophagic vacuole to lysosomal degradation process. And the whole goal is to basically bring these trucks and these recycling centers together to really kind of affect some change in there. And I'm going to just go show you one more slide. And this is actually one of the drug treatments that we've started to look at. And we've called this WICKED. And it sounds like a cool name, but it's really just the amalgamation of initials from myself and my colleagues. So it just happened that we form a WICKED team. And what we want to do is, in a kind of dose-response way, is you increase the amount of drug. Can you decrease the amount of tau protein? And we can also tag it with a red tag so we can look under the microscope as well as screen for it, as well as these aggregates. So we know that these aggregates are starting to disappear. So some of the compounds don't work. Other ones seem to be very effective in kind of reducing these tau proteins. And so our goal is to kind of take these into further development, identify some of their specificity, so we kind of challenge them with inhibitors of the same enzyme, and kind of identify potentially drugs that we can work with to get into the brain, that we can start to treat diseases. The next steps for what we want to do is take it outside of the cell, take it into an animal. Does it work in an animal model of tauopathy? And we have quite a few of them in our lab to do this. And the other part is the challenge of a lot of drugs is the brain is a very isolated environment, and it's meant to be that way to protect it. But sometimes that can be actually the physical barrier for drug development. And so what we're trying to do is also make sure that these drugs that we're developing not only can work in the models that we have, but also get into the brain cells that the brain that's really where it's going to have an effect. So the road is a little bit longer than I'd like it to be, and I'm sure the audience would like it to be, but we want to be very careful that what we're doing is hitting the right target, and that target is also going to help reverse some of the disease process. So with that, finished. Thank you.